Okay, this will serve as your fourth lecture, I believe. It is on the schedule. It's called Animal Architecture and Ecology. So this is a combination of materials, I believe, from chapters 2 and 3. These are things that I feel like you will come back to in the rest of the semester. But there's really not enough from each of the chapters to have a lecture on each chapter, so we're going to combine them here. Okay, so we'll start with ecology, and really one of the most important ideas in ecology is this idea of a niche, or some people call it a niche. And a real technical nerdy definition here is the unique multi-dimensional fingerprint of a species. And at first, this makes no sense. But I encourage you to read up on this idea of the niche and think about it. And I think at the end, the definition will actually make sense. So the way I think of a niche is the the role of an organism or an animal in its environment. It's where the animal fits into its community and its ecosystem. It is kind of like the job of the animal. Okay, and so these things are defined by a bunch of different dimensions, hence the multi-dimensional aspect. So take a given fish, for example. If you go, excuse me, to Lake Mullinex out back here, you're going to see there's a whole bunch of different species of fish that live in there. There may be up to 20 species of fish that live in that lake. Each of the different species has different niche. Okay, so each one eats something different. And in this diagram, the dimensions that we're using to define the niche are temperature of the water. Some fish like cold water, they may stay deeper. Some species like warm water, they may live in the shallow part of the lake. Salinity is percentage of the water that is comprised of salt. This is not really probably applicable to our lake here, but say in an area near the beach, this could be more relevant. And then the pH of the water. Okay, so these are just three dimensions that could define a species niche. Out in Lake Mullinex, it may be the temperature of the water. It may be how close to aquatic vegetation they live. It could be whether they eat insects or they eat other fish. It could be the depth in the water column that they utilize. All these different dimensions define where the species fits into its environment. Okay, and these dimensions are largely caused by genetic differences. So a species preference for these dimensions is determined by its DNA. Okay, and that we're going to see that fits in with morphology. So really this is actually very relevant to the discussion of the anoles, right? So a twig anole uses twigs. They eat the insects that are found on those twigs. They likewise have a unique morphology that's related to its genetics. Okay, so the niche here, we'll come back to this, but each species has its own niche. Okay, and the reason that they have their own niche is believed to be in order to avoid competition. Okay, so two species cannot occur in exactly the same niche indefinitely because they will compete with each other. Okay, so if we look at this top figure here, figure A, and we talk about anole species living in a Cuban rainforest. If these species, say for example, the height on the tree, if one species lives low on the tree, another species lives high on the tree, but they have this huge area overlap, right? That's going to be the area where they are going to be competing for sunlight. They're going to be competing for the food source. They're going to be competing for water, right? And those individuals in that population that occur in this area of competition are not going to be well suited, and they are going to have less resources and they will have fewer babies, right? And so this part of the population, and again, assuming this is genetically determined, if you have the genetics that put you in this zone, you are going to be at a disadvantage compared to relative to organisms in this zone. Okay, and so what do we see through time is that this area is eliminated and we have divergence, right? And so the niches get less breadth, so they get narrower, and the area overlap decreases. Okay, this is a very, very common um, pattern, and it's believed to drive a lot of the evolutionary change that we see. Okay, so that is, the next chapters are gonna be about natural selection, but you have some background in natural selection from 120, and so, again, this is just the basic idea. If your genetically determined trait puts you at a disadvantage, then you will have fewer babies and your genetics will be less well represented in the future. If your genetics put you at an advantage, right, then you're going to have more babies and your genetics will be more common in the next generations. And that drives the change in genetics over time. Okay, so here's an example, a real world example. Niche in these organisms, these are Galapagos finches, and these are something that Darwin really was inspired by, looking at this community of finches and looking at the patterns that he saw on these different islands. Okay, so niche in finches is largely determined by diet. Okay, and then diet is what kind of food is available to you is determined by your beak. Okay, so if you have a big thick beak like this species, I'm not sure what the genus is, but the species is Magnerostris, magna being big, rostris being nose, so it has a big rostrum, big beak. Fortis and Fuligrosa, it's kind of hard to read that, Fuligrosa. So here's the three species. We see that these guys probably eat smaller grass seeds, these guys eat heavier, thick nuts to crack. And on these islands where they all occur together, here we see the kind of partitioning of the niche. So these guys, and again on these islands too. Okay, so where these all three occur together, there's not a lot of overlap. Okay, why? Fortis eats medium-sized food, Magnerostris eats the big food, Fuligrosa eats the smaller food, and we don't see overlap. On these two islands, Charles and Chatham, again we see largely the same pattern here, even with the absence of Magnerostris. But on the island of Daphne, where only Fortis lives, we see that the niche has shifted, right? When released from competition from Fuligrosa, the niche shifts down to smaller food size. And on the island of Crossman, when only Fuligrosa occurs there, it actually shifts up, right? So when they don't have to compete with their big brother here, Fortis, they are released from competition and natural selection allows them or leads to a change in the genetics, right? Classic evolution, niche, competition, it's all there in this story. I love that story. Okay. Okay, so why ecology, right? We're about to talk about evolution. Ecology is basically the relation of the animal to its organic as well as inorganic environment. So this is where the animal fits in with all the other animals in the environment, all of the protists, all of the plants, all of the parasites and diseases and competitors and prey and commensals, and with the inorganic parts of the environment such as temperature, water, light, all these things. So ecology is sort of the distribution and abundance of organisms and what determines it, right? Now, why ecology and evolution? And no, evolution, we can define evolution. Again, that's the next couple chapters, is about the change in the gene frequencies at a level of the population. So not an individual changing its genetics, right? We know that doesn't happen. This is about the composition of a population, whether more finches in a population have little beaks or big beaks. That's a small example of evolution, right? And that changes over the span of generations. So one of the probably the most important agents, the mechanisms by which gene frequencies change, is called natural selection. And natural selection is driven by ecology. Okay, so ecology and evolution are inextricably linked, and really ecology is what drives the environment, is what drives a lot of evolution. Not all evolution, but a lot of evolution. Okay, animal architecture and complexity and evolution. It's all in this here too. So this at the top of the figure this will be on the test. So you guys got to pay attention to this one, right? Parazoan. This is before animals. These are some type of protist. This looks like a euglena or something like that. It exists, so not in, this is microbiology, not zoology. This organism exists at what we call a protoplasmic level of complexity. Okay? And what that means is they are organized, certainly. This is not a chaotic system. They have little cilia and vacuoles, and they have, DNA and all this stuff. But the protoplasm and the organelles are organized into a cell. Okay, and that's the end of the complexity. At the cellular level of complexity, we have multiple cells. This is, I think, is called a volvox. These are multiple cells living together in association. Okay, but the cells are largely independent of each other. The cells are not feeding each other nutrients. They are not communicating with each other per se. They are multicellular, but the cells are not organized into anything above that level. Okay, compared to something like sponge, within which the cells, multicellular certainly, and there are certain cells that organize into tissue types, right? So tissue is a collection of specialized cells that all work together to achieve the same kind of job. Okay, but the tissues don't organize into the next level of complexity. So the complexity ends there with tissue. Tissue organ, okay, cells organize into tissues. Tissues organize into organs, right? So we see things like organs like eyes, reproductive organs, even some very early stages of excretory organs, right? But those organs are independent of each other and they're not working together in an organ system. Okay, that is at the highest level of complexity, which, by the way, most animals occur at the organ system level complexity. And not surprisingly, the earliest animals, like sponges, occur at the cellular level of complexity. Then we get to the radiate animals that occur here, and then so it's not a hard fast rule, but as a pattern through evolutionary time, the level of organization, it has increased, okay, until we occur here at the organ systems. Cells organize into tissues, tissues work together to form organs, and then the bladder and the kidney and the urethra all work together to form organ system. The heart and the capillaries and the veins all work together to form a circulatory system, all of which are controlled by the endocrine and nervous system. Okay. So again, we got to be careful about saying that these are rules, but maybe a rule of thumb is that if we go back to the first living thing, which actually occurred pretty quickly after the Earth was created, the bacteria were formed. Um, they were and are very small. Then as we increase in time, then we have cyanobacteria, eukaryotic organisms, some type of arthropod, vertebrates, right? We get into the tetrapods, amniotes. So we see this new forms being uh, coming into existence here as time goes on. And this is not to say that the bacteria don't exist here in the present, but this is talking about the very biggest animal on Earth. So probably the very biggest animal to ever have occurred on Earth is alive today, and that is the blue whale. Okay, and so we see that the maximum body size. You have to be careful about defining what your terms are. We're not saying that probably the average body size may have increased. The minimum body size has not increased, right? Because all these things exist, but we have these new forms. So the question is, in terms of architecture and ecology, and if we see this pattern, that begs the question is, what is beneficial about being bigger, right? Why have these, as time goes on and as evolution acts on existing genetic code, why have we seen an increase in the maximum body size? And what are the implications of that? Okay, who cares? That's a good question. Okay, so that gets us back into some cellular biology. Okay, and this is kind of heady stuff in a way. So we know, and I know you talked about this in your first semester too, in 120. We're talking about size of cells. Cells are small, okay? As we increase in body size, we don't increase in cell size. So a dinosaur is not bigger than a trilobite because it has bigger cells. It just has more cells, right? And those cells are more coordinated, more carefully coordinated. Cells cannot get to beyond a certain size because of this physical relationship described on this slide. Okay, and what we see basically is the volume of a object increases much more rapidly than the surface area as its size increases. Okay, so bear with me. As the size of an object increases, so take in fact, what I've got here, I've got these little objects. We have these little sponges. Okay, I have a little sponge, and then I have a medium-sized sponge, and then I've got the large sponge, which is basically two medium-sized sponges bound together. Okay. As we go from this little sponge to this big sponge, the volume of sponge increases much more rapidly than the area of the sponge. Okay. And this comes down to this equation here. Volume increases in a cubic fashion. So the radius is the, from the middle out. So that's more of like a measure of the length, something like that, of the sponge. As this increases, the volume increases in a cubed fashion. As the radius increases, the surface area, because it's only a two-dimensional measure, increases in a squared fashion. Okay, so basically what you find is that for a given object, here the relative surface area of this small sponge is very high. So compared to the volume inside of the sponge, the surface area is very high. Okay, and getting back to functioning of organisms and ecology and evolution and all that, the surface area, say these are little tiny cells, little plant cells or something, what if the surface area would be comprised of um, cell membrane, right? And what is the importance, significance of that cell membrane? Anything that has to come into the cell is absorbed through the cell membrane, right? So sugars, oxygen, water, all that stuff that is outside of the cell that needs to get into the cell comes in through the surface area, okay? When the cell metabolizes sugars and produces carbon dioxide, any other kind of waste, and it needs to get rid of the waste, it has to get rid of the waste through its surface area, okay? So surface area is what brings things in and out of a cell, okay? The volume, on the other hand, is what is creating the demand, because inside of the cell is the protoplasm, it is all the organelles that are requiring the glucose, they're requiring the oxygen, right? So you can think of the volume is the demand, the surface area is what provisions that demand, okay? So we know that cells are very small. Why? Because they need to have a very large surface area in order to provide for diffusion, osmosis, and transport to keep up with the metabolic demands of the volume, okay? So the small size, the small radius is related to requiring a large surface area. So lots and lots of plasma membrane to absorb and to transport and to export things, okay? So this gets back to the issue of, really this gets us back to here, right? So this bacteria, does bacteria require oxygen? Most do. Does let's say first eukaryote, some kind of little amoeba or something like that. Do they require glucose? Absolutely. They have cells, they have mitochondria, they use that glucose to produce ATP. Where do they get the glucose? They absorb it through their skin, through their cell membrane, okay? Because the amoeba is small, it has enough surface area to supply its volume, okay? When you get up to something like a whale, the whale, because it's so large, simply does not have enough surface area to absorb all the glucose and oxygen it needs to provision all the cells in the very middle of the body. It has too much volume to be provisioned by the surface area, okay? A lot of amphibians are small enough that they can breathe through their skin, so they have enough surface area to supply their volume, right? But when you get up much bigger than these guys, right? And even for the amphibian, they can't absorb the nutrients. So what happens is these systems, this increasing level of complexity, essentially brings the environment into the individual cells, okay? So how do we get oxygen to our cell? We breathe it in through our mouth, through our trachea, down to our lungs, through our bronchioles, into our alveoli, absorbed into our blood. The red blood cells have an affinity for oxygen, and then they carry it to the individual cell. So we bring the environment. That's what a circulatory system, respiratory system does, is it brings the outside to the individual cell, okay? You don't need that if you're tiny, because you can just simply do that with your skin, okay? So I just think this is very interesting to think about a body in this different light, and why do we have this increasing complexity, okay? And it all comes down to these two individual equations here. Put some thought to that. Okay. This is good because that means we don't have insects that are big enough to just walk over and chomp and eat us, right? Insects don't have a complex system for providing their cells with oxygen. Here's a caterpillar that has this type of mutation where its cuticle and insides are basically transparent, and so we can see this system. Insects don't breathe in oxygen, and they don't have respiratory pigments that bring their oxygen on their blood cells through their circulatory system. In fact, they don't have a circulatory system like us. How do they get oxygen? But yet their cells need oxygen. So they have this different system called the tracheal system. There's little spiracles, little holes. You can see them here on the outside of the insect. And the air, there's a little bit of pumping. It's not just a totally passive system. The oxygen comes through the trachea into smaller and smaller branched tubules, and the oxygen is brought to the individual cells here. Much more passive system than the system that we have for delivering oxygen. So, because it's very passive and it's not very complex, you can't get very big insects. Okay? The volume of the organism is too much for its surface area. Okay, other implications of body size. Okay, large-bodied animals have a greater thermal inertia. So in other words, inertia is basically that thing that keeps an object in motion. If you're warm and you have more thermal inertia, you're going to stay warm longer. If you have less thermal inertia and you go outside, you have heat in your body when you go outside, but as soon as you go outside, your body temperature changes more rapidly. Okay. And again, this all goes back to the surface area. Okay. Because if you're big, then you have less surface area relative to your volume. The volume is the mitochondria that are making the heat. The surface area is your skin that's losing it to the environment. Okay. We add into that ecto and endotherms. Ectotherms, heat coming from the outside. Body temperature of these organisms, these are quote unquote cold-blooded animals. Things like fish, amphibians, reptiles. They generally, the body temperature is more similar to the environment, right? And they can't live in extremely cold environments, okay? Because they don't generate their own body heat. They rely on the temperature, the heat from their environment. So if they live in very cold places, there's not enough heat to be absorbed by their body to make their body function right. Endotherms, like us, right, we keep our body temperature nearly constant, but we use a lot of our food, right? This comes back to the second law of thermodynamics. When you're converting chemical energy from your food into ATP, it's not a perfect conversion. You lose a lot of heat. That heat is what keeps you warm. That's your body heat, okay? So we use tons of our energy, right? So our dogs and our goats and things at home, these days we have to feed them extra because a lot of their heat is going to keeping their body temperature up, okay? So how does this relate to body size in animals, right? We're working up to it, okay? Body temperature, a lot of that comes down to, in maintaining a proper body temperature, a lot of it comes down to chemistry. A lot of it comes down to biochemistry. Why are fevers so dangerous? Because if they unfold your enzymes, your body shuts down, right? For something like a lizard, probably not active right now today. Why? Because they have not absorbed enough energy, heat energy from the environment to make their enzymes work, right? So there's a perfect spot where enzymes work in temperature. So ectotherms need to heat themselves up during the daytime, right? They can reach functional temperatures faster with smaller body sizes. So on a day like today, a small lizard can get up to body temperature more quickly than a big lizard. So you're likely to see almost any day of the year in Georgia, you can go out and find some type of vertebrate animal, right? And those smaller animals have more surface area. They can absorb more heat more quickly. On the other hand, if you're a small endotherm, then you lose heat very quickly, right? If you're big-bodied endotherm, you lose less heat than small-bodied. So if we go to Alaska, right, and you look at something like a white-tailed deer, they're much more likely to be a lot bigger than they would be in Texas or Florida. Go to Florida and see a key deer. These things are like the size of a dog. Go to Manitoba or even to northern New York where I'm from. Deer are very big. Why? Because it all goes down to this. Again, you have more volume per surface area, so you are more likely to retain your heat than a small deer, which if you're in Florida, you actually want to lose heat, so being small is beneficial, okay? So if you're constantly losing heat, then you need to burn more food, excuse me, in order to maintain your body temperature, okay? So generally speaking, small-bodied endotherms have higher metabolism. Why? Because as they burn their food and produce heat, they're losing that heat to the environment more rapidly. Okay, this is fundamental stuff, you guys. And to me, if you want to go into zoology, you got and you start thinking about things in this manner, ecology, behavior, a lot of this stuff makes a lot more sense. Okay, this is too cool, right? Okay, here we have this. Every time you see a graph go through this process, what are the axes? One, we have body mass. Small animals, big animals. Here we have metabolism. This is calories burned per gram hour. So this is how many calories, right? How much heat you are releasing from your food, how many calories you're burning per gram of body mass per hour of time. So this standardizes how much your metabolism standardizes your metabolism for your size and for time. Okay. So standard measure of your metabolism over body size. And what we see is a very large animal. Well, we see a few things. There's a couple of stories in this figure. One is generally this trend, that as body mass goes up, metabolism goes down. Okay, so the biggest mammals, something like a elephant, has a very low metabolism. They don't burn a lot of calories per gram per hour. Okay, same thing with birds. Lizards and reptiles and salamanders, we do see the same type of relationship, but have a very different slope. So the implications of getting smaller are a lot less for these ectotherms than they are for endotherms. And just as a general rule of thumb, I mean, we look here at a given body size, 10 grams, right? Ectotherms burn so much less in terms of their food energy. So for a eutherian, 10-gram eutherian mammal burns 10 calories per gram hour. A lizard burns maybe 10 times fewer. So the metabolism is a tenth of a given mammal, and even a 20th of the metabolism of a bird. Okay, that's why you don't need to feed snakes and lizards very much. They have very low metabolism. And what else do we see here too? This is lots of stuff on this figure. Here is basically the smallest body size achievable by a mammal than by a bird. Why is that? Because if you extrapolate back here, the mammal, a 1 gram eutherium mammal would need to burn 20 calories per gram hour, which is basically if you eat Crisco and biscuits, sausage biscuits all day long, you simply can't provide enough calories. Okay, so what is the tiniest vertebrate? Always, right? We'll follow the link here. Maybe it'll work. Keep your fingers crossed. What do you suppose? What is it going to be the tiniest vertebrate? Is it going to be a amphibian, a reptile, a mammal, or a bird? Well, yeah. Let's see real quick. Oh, forget it. I hate these computers. It's pulling it up. What is this? Yeah, a fly-sized vertebrate. Look at, here's an American dime. This is a full-grown adult Paedophryne amuensis. And okay, yeah, so it's a frog. Damn it. Okay. Salamanders tend to be a lot smaller. Lizards tend to be a lot smaller. Mammals tend to be bigger. Probably exactly for these reasons. Okay. Biggest animal on Earth, the blue whale, a mammal. Okay. There are benefits to being so large. You can have very low metabolism, right, which is beneficial. You can't be eaten by much, right? This is about the smallest mammal you can possibly have compared to that really small frog. Okay. So body size, very, very important. But notice that smallest vertebrate lived in the rainforest where there's lots and lots of water. A small frog is at the mercy of the elements because they lose water to their environment very quickly. So we go back to our little sponges here. What I did to these sponges before I started this lecture, all I did was wet them, I wrung them out, I measured their mass before we started. Okay. What I'm going to do is I'm going to look and see, should be, relatively speaking, which one of these should have lost the most water. All right, think about that. Relatively speaking, should the small sponge have lost more water or the big sponge lost more water? Not total amount of water lost, but relative to its size, which one should have lost the most? Right? Which one has the highest relative surface area? So what I'm going to do is I'm going to remeasure these things. This is just half an hour later. This one is now 2 grams. This one is now at 10 grams. So they both lost a half a gram of water. And then this chunky boy (22.5). So they all lost half a gram. Okay, but half a gram is what percentage of the total? So we'll see here that 0.550 100 times 0.5 is this. So this one lost 2.17%. This one lost 5% of its body mass in water, and the little one lost, let's see, 0.5, 50, 20%. Okay, this little guy lost 20% of its body mass. So that's like saying if you weighed 100 pounds, then you lost 20 pounds of water. This guy here lost only 2% of its body mass in water over the course of this lecture. Right? Huge implications. Why would you want to be a big frog? Well, it's really easy to stay hydrated. If you're a tiny little frog, you're screwed if you're in a really dry environment because your surface area is so big and you're losing water constantly. Okay, so that is about body size, niche, evolution, all right, so on and so forth. Last part of the lecture, I'm going to go through this pretty quickly. A lot of this stuff in the last part of the lecture is just you have to learn this on your own. Okay. Now, this is a big phylogeny. At first, it doesn't really look like a phylogeny, but what is it? It's a branching diagram that shows the relatedness of these different phyla of animals. Here's arthropods. Here we are in the mammals. There's birds, reptiles. Here are things like flatworms. Here are things like nematodes, tardigrades, starfishes, right? We're going to learn about most of these big ones, annelids, mollusks, nematodes, arthropods. We're not going to learn about these little minor phyla. Okay, but a big part of the rest of the semester is just breaking this down and looking at the interrelationship of these major phyla and looking at what characters go on this phylogeny that help us to make sense of the diversity of all these animals. Okay, so the next couple slides we're looking at laying the kind of background foundation in terms of the characteristics that are important that go on to this phylogeny. Okay, what are those characteristics? One is symmetry. This is looking at the shape or pattern of the animal body. We can talk about asymmetrical animals. These don't have any symmetry at all, right? These are things like a sponge. Why is that beneficial? Well, if you live in an environment where there is no predictable pattern as for where resources are going to be found, then you're not constrained to grow in a symmetrical fashion. If there's space over here, you can just grow and absorb that or use that space. If there's light over here, you can grow in that direction. Okay? Spherical symmetry is basically a sphere, and if you pass a line through the middle of the organism in any plane, you're going to get two holes. So it's basically a ball. Slice a ball in half, you get two balls that are identical. Okay? Not really any animals in this. This is beneficial if you're tumbling or rolling around in an environment. All right? Some other animals, though, we have radial symmetry. This is more like a pizza pie. You have flattened on one plane, but round in the other plane. Jellyfishes, sea stars, anemones, things like that will have radial symmetry. And this is good if you are either attached, sessile, not moving, or you move in a slow manner and not directionally. So you're moving this direction, that direction. You're planktonic and you're sensing on your back and your front. There is no front or back, right? Because you're not moving in a given direction. As opposed to bilaterally symmetrical organisms. They have two-sided symmetry. So if you cut the organism from front to back, because they do have a front, they do have a back, you're going to get equal sides. Okay? So most animals are bilaterally symmetrical, including us. Oh, perfectly symmetrical. One of my legs is a lot longer than the other. but if you pass a line, then both of the sides are more or less symmetrical. Okay, that is for animals that are sensitive to their environment, they're motile or moving, right? And usually these animals have a head end. That is to say, they display cephalization. Cephala means head, okay? Cephalization, right? Concentration of sensory structures anteriorly. The anterior, posterior. Anterior end of the organism has the eyes, the sense of smell, usually whiskers, and lots of sensory structures. Why? Because as they move into a new novel environment, they want to know what's going on there. Okay, and so that's where the sensory structures are concentrated. So radial symmetry, pizza pie, slice of pie, two equal sides. Now it's not spherical in that there is a top side and a bottom side, but maybe just not a left and a right side. Bilateral symmetrical, right? Do learn these planes. We've been talking about the sagittal plane here, but this is a good figure to know. Dorsal, top, ventral bottom, anterior front, posterior back. Right? You can have a transverse plane where you slice the organism this way, sagittal plane, or a frontal plane. Frontal plane. Again, know these. Just I can explain them. These are really easy to understand. You just want to know these as just the basic vocabulary of biology. Proximal, say you're talking about where feathers are attached on a wing. If they're proximal, then they are at this end of the appendage towards the attachment point. Distal, think of they're distant, they're out on the tip of the appendage. Okay, segmentation, some organisms, some animals are segmented. They're metameric, right? So, for example, things like millipedes and centipedes are have really highly developed metamerism. Okay, and then let's see. Body cavity is another important trait used to differentiate organisms, whether they have this fluid-filled cavity called a coelom. You can be acoelomic, so no coelom, no cavity. You could have a true cavity, a eucoelomic organisms, right? And we're going to see that these the coelom gives organisms an advantage because it helps with circulation, right? This would be like our peritoneal cavity is our coelom, and that is where a lot of our body fluids are concentrated. Some organisms, very primitive animals like the flatworms, have no body cavity at all here, but a lot of animals do have this true body cavity. And then finally, this is one more just terminology type thing that you should know on our big phylogeny. Protostome, deuterostome. Okay, I stress this a lot, this etymology or the meaning of words. You want to break these words down. It's going to be on the test. I promise you, you're going to be asked to differentiate between these two. If you just memorize this like a pigeon learns to read, just looking at this as a symbol, you're going to be more likely to forget it. Now, break it down. What do we see in these two words? Proto and stome. Stome, what does that mean? The stomata on the underside of a leaf. They're like little mouths. Think about the stoma. Right? People that have smoke too many cigarettes have to have a little stoma cut. Stome means mouth. Proto means first. First mouth. Deuteronomy is the second book. Deuterostome is second mouth. So as a animal develops, right, it at some point develops into a multicellular hollow ball of cells, right? And that invaginates or dimples in, and that pore, the blastopore that forms, continues to grow, and it forms a tube, right? And that's basically the most basic um, way to describe an animal body is a tube within a tube. The tube is the mouth through to the anus. This is the gastrointestinal tract. And it forms very early in development. The blastopore dimples in, grows through, forms a tube. If that blastopore later, the first opening is the mouth, then they are protostome. If the first opening is the anus, and then the second opening is the mouth, then you are deuterostome. Okay, there are some other characteristics of protostome, deuterostome. This goes a lot more deeply. It depends on whether as cells cleave during mitosis, if they cleave in a spiral manner or radially, right? There's a lot of other things that are relevant, but just to know, all right, so I'm going to go back to our big phylogeny here. Here it is. So we see one of the most fundamental splits in the bilaterally symmetrical animals is between this group where we find ourselves and this huge group where most of the other bilaterally symmetrical animals are. Protostomes, deuterostomes. Did your mouth develop first or did your mouth develop second? Okay, that's all there is to it. Okay, well, I'm coming up on an hour here. This is way too long. Watch us and break it up into 10-minute sections if you want to. All this stuff is important. Ask if you have questions, and we'll see you soon.